Staphylococcus organisms are gram-positive cocci that appear in grape-like clusters. The most clinically important species is Staphylococcus aureus, which is coagulase-positive and the most common pathogen of the genus. It causes infections such as furuncles, carbuncles, bullous impetigo with blisters, paronychia of the nails, and post-surgical wound infections that may lead to bacteremia. It also produces intoxications. In neonates, exfoliatin toxin causes scalded skin syndrome. Toxic shock syndrome, associated with TSST1, is especially seen in women between the ages of 12 and 52. Food poisoning results from ingestion of enterotoxin, with symptoms appearing within 1 to 5 hours, often linked to foods like potato salad or cream dishes. S. aureus produces many exotoxins, including hemolysins, leukocidins, coagulase, hyaluronidase or spreading factor, nuclease, protease, and lipase. In terms of resistance, most strains are resistant to penicillin due to plasmid-mediated beta-lactamase. Some remain sensitive to penicillinase-resistant penicillins such as methicillin or oxacillin. Methicillin-resistant Staphylococcus aureus, or MRSA, is best treated with vancomycin. In the laboratory, colonies on blood agar appear soft, opaque, and about 2 to 3 millimeters in diameter. Some are beta-hemolytic, and some show a pale golden color. The organism grows in 7.5% sodium chloride and ferments mannitol. It is catalase positive and coagulase positive. Coagulase-negative Staphylococcus are generally opportunistic, especially in immunocompromised hosts or patients with prosthetic valves and devices. They are catalase positive but coagulase negative. Staphylococcus lugdunensis is unique in being PYR positive and ornithine decarboxylase positive. For identification, we use biochemical tests. The catalase test employs 3% hydrogen peroxide. A drop is added to the colony, and if catalase is present, bubbling occurs as hydrogen peroxide breaks down into water and oxygen. Staphylococcus is catalase positive, while Streptococcus is negative. Daily quality control is required. The coagulase test uses EDTA rabbit plasma. Bound coagulase is detected by clumping on a slide. Free coagulase is detected in a tube test, which gels after 4 to 12 hours at 35 to 37 degrees Celsius. Agglutination tests can also detect coagulase and protein A. A positive result indicates Staphylococcus aureus, while other human pathogenic staphylococci are negative. Imagine Staphylococcus aureus speaking to you in verse, claiming its place in the Pathogen Hall of Fame. It says, "I am known as Staph aureus. Look for me in grape-like clusters when examining gram-positive cocci. You can detect me with the catalase test, where bubbles form, and with the coagulase test, where clots appear." It reminds us that it can be found everywhere, on the skin, in the gut, and in the nose and hair. Its virulence is tied to protein A, which interferes with the immune response, and to its enterotoxin, responsible for food poisoning. Staph boasts of its resistance. Penicillin doesn't work due to beta-lactamase production. Methicillin resistance is common, giving rise to MRSA. And while vancomycin used to be reliable, today even that drug faces resistance challenges. The rhyme ends with a cautionary note. Wash your hands, don't pick your nose, and wear a lab coat over your clothes. This underlines the importance of infection prevention, hygiene, and protective measures when dealing with this notorious pathogen. The genus Staphylococcus is catalase positive. 3 important species stand out in clinical practice. Staphylococcus aureus is coagulase positive and the most virulent species. It causes infections such as carbuncles, furuncles, paronychia, wound infections, and bacteremia. It also produces intoxications, including scalded skin syndrome, toxic shock syndrome, and gastroenteritis due to enterotoxin, with symptoms appearing as quickly as 1 to 5 hours after ingestion of contaminated food. Most strains produce beta-lactamase, and many are methicillin resistant. The MRSA strains. Infections may be community-acquired or hospital-acquired. Staphylococcus epidermidis is coagulase-negative. It is a major cause of endocarditis and infections associated with prosthetic devices. Most strains are resistant to methicillin but remain sensitive to novobiocin. Staphylococcus saprophyticus is also coagulase-negative. It is best known for causing urinary tract infections in young women. Unlike S. epidermidis, it is resistant to novobiocin. The key point is that coagulase-negative Staphylococci are opportunistic pathogens, particularly in immunocompromised patients and in those with prosthetic devices. Streptococcus species are spherical or oval bacteria, typically arranged in chains or pairs. They are classified by Lancefield grouping, which is based on their C carbohydrate antigen. Streptococcus pyogenes, or Group A Streptococcus, is beta-hemolytic. It produces 2 hemolysins, streptolysin S, which is stable in oxygen and non-antigenic, and streptolysin O, which is oxygen-labile and antigenic. It also produces erythrogenic toxin, which causes the rash of scarlet fever. Group A strep is highly sensitive to penicillin. It causes pharyngitis, impetigo, erysipelas, wound and burn infections, and can lead to rheumatic fever as an autoimmune complication. In the lab, it is identified by its sensitivity to the bacitracin disc and can be further typed serologically. Streptococcus agalactia, or Group B Streptococcus, shows a narrow zone of beta hemolysis. It is a leading cause of neonatal sepsis and meningitis, and is also associated with urinary tract and vaginal infections. Laboratory identification includes serotyping, a positive CAMP reaction with Staphylococcus aureus, and a positive sodium hippurate test. Group D Streptococci include 2 major groups. Streptococcus bovis, also called Streptococcus galolyticus, is bile escalin positive but does not grow in 6.5% sodium chloride. It is associated with colorectal cancer. Enterococcus species are also bile escalin positive, but do grow in 6.5% sodium chloride. They cause urinary tract infections, bacteremia, and other opportunistic infections. Streptococcus pneumoniae is alpha-hemolytic and produces colonies that may appear crater-like or mucoid and watery. Microscopically, they are lancet-shaped diplococci. Penicillin sensitivity is tested using an oxacillin disc, where a zone of 20 millimeters or less suggests sensitivity. Clinically, it causes primary lobar pneumonia with rusty sputum, meningitis, otitis media, bacteremia, and conjunctivitis. Laboratory diagnosis includes recognition of its colony morphology, the quelling reaction, optokin sensitivity, and bile solubility. Streptococcus pyogenes is a significant human pathogen. It remains highly sensitive to penicillin, which is the treatment of choice. Its major virulence factor is protein M, which interferes with immune defense and contributes to throat and muscle infections. The anti-streptolysin O antibody is associated with post-streptococcal complications such as damage to the heart and kidneys. In the lab, Group A Streptococcus is recognized by its sensitivity to bacitracin, where a clear zone of inhibition confirms its presence. On blood agar plates, S. pyogenes shows beta hemolysis, completely destroying red blood cells around the colonies. This organism highlights the importance of laboratory diagnosis, recognition of virulence factors, and effective antibiotic treatment. To remember the difference between staph and strep, think about their arrangement and key tests. Streptococcus organisms are arranged in pairs or chains. They are catalase-negative, meaning no bubbling occurs with hydrogen peroxide. Some, like Enterococcus, can grow in 6.5% sodium chloride. Staphylococcus organisms appear in grape-like clusters. They are catalase-positive, producing bubbles with hydrogen peroxide. They can grow in 7.5% sodium chloride. Importantly, Staphylococcus aureus is coagulase positive, which distinguishes it from other staphylococci. This simple framework helps you quickly separate the two major genera in the lab,